My name is Adrian Vasquez, and I'm a biologist, and I work at a university in Detroit, Michigan. One of the main things that I do is that I study biodiversity. The way I study the diversity of biological animals is that I go out into the field, I go out into the lakes that are found here in Michigan, the Great Lakes. I go out to Lake Erie or Lake Huron or Lake St. Clair or here in the Detroit River, and I use nets. I use different types of sampling equipment. I put on a waders and I get into the lake and I stir up the bottom of the lake and I use the net to draw it across the plants that are found in the lagoons and lakes. And then I take that sample and I sieve it in a sieve just like you would sieve some vegetables in your kitchen. And what happens is I trap all the little invertebrates that live in the lake. And then I bring those back to the lab and I use microscopes and other viewing devices to see what those things are, because they're microscopic. You cannot really see them properly with the naked eye. And then I study those and I try to identify them by their morphology, by what they look like. And these animals are from all sorts of different types. I also work with DNA of these animals. So I use technology that I can extract the DNA from these animals and that I can do what we call sequencing. We get the sequence and then we are able to identify them further genetically. And this way we can give you a picture of the diversity of biological organisms that are found in any given place that we sample. After that, we consult with other experts. We work with other scientists from across the world. These scientists collaborate with us, help us to be able to further identify these animals and tell us more about their life habits and the concerns or the issues that we have with them. And then we would write a story about that, about any particular thing that we discover that is of interest. And we would publish those stories in journals that are relevant to the area where we work at. And then we use our methods to work with students. We would invite students here to the aquarium or to the Metro Park. We would show them these methods that we use, and they would sometimes assist us in collecting these organisms. And they would have a lot of fun because a lot of times when they collect these animals, it's probably the first time that they are being studied in science. So they might be new species which they themselves can then name and put a name on these new animals that are being brought to science. This kind of work is occurring around the world, but with larger organisms. We are focused with the tiny ones. We are focused with the microscopic organisms, and we consider them charismatic, just as you would consider a toucan or a scarlet macaw. We would consider these tiny little critters as charismatic, as beautiful, and as very important because they are found at the bottom of the food chain. They are what sustain the entire system. It's necessary to study them and to understand the roles they play and the potential issues that can come when these critters are affected by different problems.